Welcome, and thanks for coming to this late afternoon session. I'm really excited to be here to hear about all of the exciting new therapies that are coming out based on engineering cells. And so actually, today I'll be presenting our focus at B-MoGen Biotechnology, which is actually a spin-out company from the University of Minnesota. And our focus is actually building new tools to genetically modify cells of interest for cancer immunotherapies and also for gene therapy. And so the title of my talk today is Non-Viral Gene Delivery Using DNA Transposases for Clinical Applications. So this is one of the main focuses of our company, is to generate non-viral gene delivery methods to essentially replace retroviral and lentiviral vectors for stable gene transfer into any cell type. And so just a little bit of background for those of you who have never heard of what DNA transposons are. So they're essentially a system that's evolved in nature whereby there's two components to the system. There's a DNA component, essentially analogous to like if you would have a lentiviral vector and you'd be able to put cargo or trans genes like CARs or TCRs in there. And then there's also an enzyme component. And so what happens is this enzyme component actually recognizes the DNA component and integrates that stably into the genome of cells. And so you can actually deliver this system through any method into cells, whether it be electroporation, liposuction, or even squeezing nucleic acids into these cells. And then once the system is in there and you have stable integration, you actually get stable transgene expression for the life of that cell. So it does exactly the same thing that a viral vector does, but it's non-viral, and therefore much safer, cheaper, easier to manufacture, and I'll get into some of those details in a minute. And so actually in nature, there's many superfamilies of these transposon elements, and many of them have actually been silenced through evolution because they can be detrimental to the development of an organism, and so they've been inactivated. But now, actually at the University of Minnesota, we reactivated a transposon system in the early 90s called the Sleeping Beauty Transposon system, so you may have heard of this. It was actually developed from inactive transposons in fish, and so a company actually licensed that technology, and we worked with MD Anderson to run a clinical trial to deliver genes to cells using this mechanism. And there's also another variant called piggyback that's being pursued for the same purposes. And in our case, we're actually studying the HAP family of DNA transposases. And so these are from a red beetle family, and specifically the system that we've developed is called TC Buster. And so that will be the main point of the talk here, is how to use this system for stable gene transfer. So interestingly, transposons can be used to engineer any cells, as I mentioned, but specifically CAR T cells or TCR transgenic T cells. So here's just a diagram of what many of you are likely familiar with, using a patient's own T cells, delivering a CAR TCR. This is typically done with a viral strategy, but in our case with a transposon, and then those cells are used to fight an existing cancer in cancer patients. So interestingly, the founders of our company and also members of our scientific advisory board, we performed the first in-human clinical use of a DNA transposon for stable gene transfer. And so in this case, we worked with Lawrence Cooper at MD Anderson using the Sleeping Beauty transposon, delivering CARs to T cells for CAR T cancer immunotherapy, and essentially saw fantastic results, very comparable with lentiviral delivery of CARs. And so as I mentioned earlier, this has been exclusively licensed to Xiopharm and Trexon and Merck, and so this is not really available for other cellular-based therapies, and that's why we're developing alternative transposon systems that are still available for use for cellular therapies. And not only can you do this for T-cell therapies, but also you can use them for many other diseases, including genetic diseases, which are just listed here in some of the kind of upcoming trials, specifically with the Sleeping Beauty transposon system. And so, as I mentioned earlier, we believe that transposons are far superior to lentiviral vectors or retroviral vectors for stable gene transfer. So first of all, they both are FDA approved. They've been tested in humans. They both have the same outcome, so stable gene transfer to have expression of your transgene for the life of the cell. Interestingly, the cargo sequences that you can package within a viral vector, they have limitations because when they are packaged, there's reverse transcription steps, and so you sometimes can't put in the sequences that you want. With DNA transposons, actually, you can put in any sequence you want because there's no packaging, it's just nucleic acids. Also, there's size limitations with viral vectors, typically 10 to 12 kb with lentiviruses and even less with retroviruses. And actually, transposon vectors can typically deliver well over 12 kb in cargo and even up to 100 kb in cargo. And this is good because you can use it to deliver multiple genes at once, for instance, like a CAR and a suicide gene for safety. So there's a lot more room for engineering with transposons. Also, viral vectors can have immunogenicity, where again, DNA transposons are basically transiently expressed, and then they just leave DNA behind, which is essentially inert. And also, there's the worry of insertional mutagenesis with lentiviral vectors. They actually prefer to insert near active genes and can drive oncogenesis. And actually, transposases, it's dependent on the system that you're using, but some of them, like Sleeping Beauty, for instance, it's a near random insertion profile, so it's actually very safe compared to a lentiviral vector. And importantly, the cost of either system is very different. So with lentivirus systems, you have to have lots of manufacturing and safety concerns, whereas with DNA transposons, you're just generating nucleic acids, which are very simple and cheap to manufacture. So it actually becomes much cheaper for stable gene transfer. And then a couple other things. So there is no DNA toxicity associated with viral vectors. In fact, very low toxicity in most cases. And one issue that has been very apparent with DNA transposons is DNA toxicity. And so today I'll show you about some methodologies and systems of engineering that we've developed at B-MoGen to completely eliminate or largely reduce DNA toxicity associated with DNA transposons, which is really one of the only drawbacks of using this system. And then lastly, the efficiency of both systems can be very high. In fact, with using mutant versions of the transposase enzyme, you can actually reach the same level of integration efficiency as viral vectors. And so with kind of the pros and cons and comparisons laid out, there has been a lot of perceived disadvantages of DNA transposons that I can mitigate with some facts. And so first of all, people think that using DNA transposons are low efficiency. And in fact, in the past, that was the case. You can see here our Sleeping Beauty transposon system we used in the clinic. The efficiency was only about 10%, whereas with lentivirus, you can get 50 to 70%. But now people have developed hyperactive transposases to reach the rates of viral delivery as well. And so in addition to that, people have developed strategies to minimize the DNA cargo of a DNA transposon because they're delivered as plasmids. So they use these mini-circle plasmids, and essentially this allows for even more increased efficiency. You can see here almost 60% stable gene transfer in T cells. Another concern is uncontrolled transposition or integration of the vector and then remobilization and integrating again. But actually, this is largely mitigated using mRNA or protein enzymes because they get turned over really fast, and so you get your integration event, and then the enzymes dissipate very rapidly. And then lastly, DNA toxicity. So here's just some data with Sleeping Beauty, and you can see in T cells here that the cell viability using the transposon system delivered as plasmid or even mini-circlin RNA, it's actually quite toxic to some cells. And B-MoGen, like I mentioned, has developed methods to overcome this. And so here's just some data showing our B-MoGen kind of proprietary engineering methods for overcoming DNA toxicity. So we compared to the standard methods for engineering primary human T cells using viral vectors. And what you can see is the B-MoGen methodology actually improves viability, it maintains the efficacy of gene transfer, and it also improves the expansion of the cells to a very comparable level to cells that are unmanipulated or just electroporated with an mRNA, which is largely inert in T cells. And so this has allowed us to develop this system that we call TC Buster, and we've generated comprehensive patents for the superfamily of transposon and transposase elements. And so importantly, our system is actually very comparable to SB11 that was used in the clinic. And so here's some just data looking at stable gene transfer and transformed cells. And importantly, when we compare it to these hyperactive mutants of Sleeping Beauty that achieve levels of gene transfer comparable with viral vectors, we actually developed a transposase enzyme variant that is just as active as SB100X, and we expect to use this for primary T cell and other cell engineering to reach the level of viral vector integration rates. And lastly, even without this variant that we've developed just with our baseline system, we can actually achieve over 20% stable integration in primary T cells as it stands right now. And so just some of the key takeaway points from my talk today. DNA transposons are an economical and safer alternative to viral delivery. Recent advancements have improved the rates of transposition, so the efficiency of gene transfer, and also methods for increasing their viability. Sleeping Beauty and Piggyback are now no longer the only transposons in the market for clinical use, so we've developed a novel system that we call TC Buster. B-MoGen has also developed these proprietary methods to almost completely eliminate the DNA toxicity associated with using DNA in primary cells. And as I mentioned earlier, this is kind of the biggest problem or perceived issue with using this type of non-viral gene transfer mechanism. And so lastly, the HAP family of DNA transposons are a safe, efficient, and cost-effective non-viral gene delivery platform that are available for clinical use through our company. And so I just also want to put a plug in for our new facility that we're building in Minneapolis, Minnesota. So we're actually building a GMP-compliant non-viral cellular manufacturing facility. And so this will kind of be an end-to-end type facility where we develop the transposase GMP, the transposon, and then also do the cellular engineering. Or we can partner with other manufacturing facilities and simply provide the transposase and the transposon in a GMP fashion for cellular engineering. And so with that, I just want to acknowledge all the key people at our company and the Scientific Advisory Board and Board of Directors. And with that, I'll take any questions that you guys have today. And thanks for your attention.